Welcome to Parasitic Diseases. Today we're going to discuss African trypanosomiasis, otherwise known as African sleeping sickness. This is a disease restricted to the African continent, and it's transmitted by a vector known as the tsetse fly. There are two species that we'll discuss, but we'll probably put them together for the clinical picture because the clinical picture for Trypanosoma brucei gambiense is a little bit different than the Trypanosoma brucei rhodesiense clinical picture, but they both have a lot in common, so we'll discuss them together today. To begin with, there are really two forms of the disease which are transmitted by multiple forms of the vector. So Trypanosoma gambiense is found in West Africa, and we can see from this map its geographic distribution. The pluses represent where the tsetse fly vectors are found that transmit this infection, not just from person to person, but it's also from person to animals as well. From this map, you can see the distribution for both Trypanosoma brucei gambiense and Trypanosoma brucei rhodesiense. The pluses represent different species of Glossina, which is the genus name for tsetse flies. Tsetse fly is a common name for the vector. You can see that the tsetse fly belt is definable by the pluses that goes across West Africa and then turns south in East Africa. The diseases are quite different in terms of both their clinical manifestations and whether or not there are reservoir hosts associated with this. So in West Africa, for instance, there are very few animals that can harbor this infection besides people. One of them is the domestic pig, which is kind of unfortunate considering how widely spread pig husbandry is throughout West Africa. In East Africa, it's a disease of people, but it's an infection of most of the game animals found throughout East Africa: elephants, hippopotami, gnus, wildebeest. Those are the same animals. And various hooved animals in East Africa, serve as reservoir hosts for this infection, which in them represents just that, it's an infection, but in people represents a very serious disease. As the result of the distribution of this one vector species throughout Africa, and remember Africa also has lots of other vector-borne diseases, malaria, leishmaniasis, for example, and onchocerciasis, which we'll get to much later in this course. The distribution of where people live in Africa is largely determined by the absence of tsetse flies. So we see, for instance, in this map, you see there's a zone above the red zone, which is where the tsetse fly belt is. And all those little black dots that you see, each one of those dots represents 5,000 head of cattle. People are tending their cattle, and they're being squeezed, so to speak, by the Saharan desert to the north, pushing south, and the tsetse fly belt pushing north from the south, and they're caught in between this zone. And that's where you find a lot of animal husbandry, a lot of people living, and a lot of transmission of other diseases besides trypanosomiasis. So avoiding the tsetse fly belt allows for cattle husbandry. And you can see, of course, that throughout South Africa and Namibia, there is no transmission zone for African sleeping sickness, and therefore a lot of cattle are raised in South Africa and in Namibia as well. So how do we know all this? Actually, it's almost due to one person's perseverance. David Bruce, a microbiologist from England, set forth to discover new diseases, as was the wont of a lot of people during the 18th century and early 19th century. And he began by investigating cattle diseases. And one of the cattle diseases that he's become famous for has his name on it, namely brucellosis. It's a cattle disease which results in spontaneous abortion in cows. After discovering the causative agent for that disease, he moved to the African continent at the request of some people living in West Africa that said, if you are interested in cattle diseases, you should come here and look at our cattle because we're having problems and we don't really know what the cause of those problems are. Of course, Bruce immediately went to West Africa and discovered the problem simply by looking at the cattle. As you can see in this very, very emaciated looking white cow, the Ndama cattle of West Africa, you can see the bone structure showing through the skin, producing very little meat. This cow is suffering from a disease that all the locals knew as Nagana. So Bruce, of course, asked, what is the cause of Nagana? And in doing his work, he discovered not only the causative agent for Nagana, which now bears his name, Trypanosoma brucei, but he also discovered that it was transmitted from cow to cow by a biting fly, which locally was known as a tsetse fly. So because of Bruce, we now know that African trypanosomiasis is a vector-borne disease caused by trypanosomes, which can infect animals. And shortly thereafter, other investigators throughout Africa discovered that it could also be transmitted to people and cause African sleeping sickness, which we will describe shortly. Let's look at the life cycle first of this organism, and we'll lump them together because basically they both behave similarly in the mammalian host. So the tsetse fly bites. It's an infected tsetse fly, of course. It injects its load of trypamastigotes. In this case, it's known as a metacyclic trypanosome. The trypanosome then enters the lymphatic vessels surrounding the bite wound and develops into a primary chancre, which takes a couple of weeks to develop. And it's innocuous, it's painless, and the organisms are multiplying within the lymph that collected in this bite site. It may even have some remnants of fly salivary gland proteins, which may help to facilitate this chancre. We don't know that, but we do know that other vectors for other infectious agents, as we discussed for leishmaniasis, there is a collusion between the parasite and the insect vector to not only aid in the successful obtaining of a blood meal from the vector, but also aiding in the parasite's success as a parasite. As this chancre, as it's now referred to, develops, and it, of course, because it's a chancre, it must be distinguished from other chancres, namely syphilitic chancre, etc. The organisms can gain entrance into the blood supply, and once they do, they are then free to replicate. And the trypamastigote stage does not invade tissues. It remains in the blood supply for a long period of time in this host. Let's make this a human host in this case, and undergoes numerous replication cycles, depleting the glucose supply in the blood supply of the infected individual. Eventually, in a matter of weeks, with regards to the rhodesian species, it's now the Zimbabwe species, we should bring that up to date perhaps, and in months, in terms of the gambiense species of trypanosome, the organism then finds its way into the central nervous system in the cerebrospinal fluid. This has grave consequences for the host, of course, and induces a stupor in the patient, which results in lethargy and hence the name sleeping sickness, because a lot of people with this infection in the late stages are unable to actually develop enough energy to even get out of bed. The cycle continues when an uninfected tsetse fly bites an infected individual, acquires the trypamastigotes, and then the cycle begins inside the tsetse fly itself, which we'll get to in a little while. Some of the early consequences of infection with people include the swelling of lymph nodes on the back of the neck, and a physician in Africa by the name of Winterbottom noticed this in a lot of individuals that A, had a primary chancre, and B, went on to develop these swollen lymphatic vessels, lymph nodes rather, in the back of the neck at the nuchal crest. And it became known as Winterbottom's sign. It's a sign that indicated that this person will go on to develop a serious infection, which, of course, is African sleeping sickness. Other consequences of the infection will be discussed when we discuss the clinical manifestations of the disease. Let's return now to where this organism lives. And indeed, if you look in East Africa, you can find virtually every animal species that's worth going and seeing on safari as a potential host for this infection. And that's why it's such a popular infection throughout East Africa. The trypanosome has a different life form in both the mammalian host and in the insect vector. And it's distinguished by the fact that the shape of the organism actually changes over time as it differentiates from one stage to the next. And then as it's taken up by the tsetse fly, it undergoes replication and differentiation into the infectious stage. And we'll see more about that in just a moment. So here we are looking into the face of the vector. This is a very large biting fly with blades that it slices back and forth in order to gain entrance into the tissue. It's a very painful bite. And people talk about how painful it was to be bitten by these flies. The animals, of course, hate them. And you can find zebras standing head to tail with their tails brushing the flies off of their faces as they mutually try to keep these flies down to a minimum. That's true for West Africa. And by the way, in East Africa, the density of tsetse flies, we'll talk about that when we talk about the control of this infection. The density of tsetse flies in East Africa is quite low, actually. It's less than 1 fly per mile of open grassland in the Serengeti, for instance. You'd think with a low density like that this would have a very small chance of success, but it's actually a very successful vector in terms of transmitting this infection from animal to animal. In West Africa, the flies live along the riverbanks as opposed to the open savanna in East Africa. And there are millions per square mile along the rivers. And therefore, the best way to avoid infection with this infection in West Africa is to live further away from the river bank than the flight range of the fly itself. And that's very possible to do. And in fact, you can find a lot of settlements in West Africa that are not right on rivers, and you wonder, why aren't they on rivers? And this is one of the explanations, of course. There are others too, namely the onchocerciasis that's present there as well. Another disease we'll discuss later. Development of the trypanosomes in the tsetse fly vector is complicated. Just like the development of Leishmania parasites in the sandfly was complicated. But we can simplify this somewhat to let you know that it's not a process that we don't understand, at least in terms of the overall process. But the nuance of this, the ligands that bind from one stage of the parasite to the next inside the gut tract of the sandfly or the tsetse fly vector in this case, are little known, and we're still exploring those possible binding sites. But what we do know about this parasite is that when the blood meal is obtained by the fly, the organisms travel all the way down to the midgut. And the long, slender forms, the bloodstream form, the BSF forms, are digested by the fly's secretions in the stomach. But one stage survives. This short, stumpy form actually survives that process and begins its differentiation in the midgut of the fly to these procyclic trypanosome stages. The procyclic stage multiplies vigorously inside the midgut, and then it has to pass through the peritrophic membrane, which divides the anterior part of the digestive tract of the tsetse fly from the posterior half of the tsetse fly's digestive tract. It's a membrane that's actually physical that these organisms have to pass through. And in doing that, it stimulates them to differentiate into the stage just before the infectious stage of the infection, which is known as the long, slender epimastigote stage. They then undergo further development to the short epimastigote stage, for want of a better term. We don't really have molecular markers for these that we could say this is the that stage and this is the stage. We just simply refer to them as their shape stage. And finally, the short epimastigote stage is able to differentiate into something which is indistinguishable from the original blood meal long, slender forms of the bloodstream forms. And at this point, this organism is now ready for life inside of its mammalian vector. Interestingly enough, as we'll see too, the outer surface of this organism is covered with a single protein, which it secretes and puts into place to protect it from host immune responses. So it's in the fly, and there are no host immune responses to speak of in the fly. It would interrupt the life cycle of this parasite, but it's already getting ready to encounter the host immune system. And that's a remarkable developmental cycle, which very few other organisms have imitated in their own biologies. And you can see some interesting abbreviations down here. We'll explain all of those when it's time to discuss what the organism does in terms of its pathogenesis. So to know about the pathogenesis of this organism, we have to know more about the trypanosome itself, because a lot of it is due to its ability to change its outer coating in response. Well, we used to think it was in response to the host immune response, but it's actually not. It's a series of timed productions of surface coat proteins, all of which are related to each other, but they're antigenically different at the site of exposure to the host. And these are actually cassettes of genes which are put into motion in the fly, but which stay in motion once it's in the host. So although you're looking at a very complicated drawing here, I mean, here's the nucleus of the organism, for instance, and the kinetoplast. Again, trypanosomes and leishmania form this interesting group called the kinetoplastidae. The undulating membrane for the flagellum is found here, and then, of course, the flagella is out here, which gives it its ability to move around. If we look closer, though, we see some very interesting things. First of all, we see where the flagella comes from. It comes from a specific site in the organism, and it's surrounded by this open region called the flagellar pocket. It's thought that most of the absorption of materials that the parasite uses to grow with, like amino acids and nucleic acids, short-chain fatty acids, etc., are all picked up through this flagellar pocket, and that is, for the most part, still true. This is data that was collected, let's say, over the last 20 years, but it's now known that the surface of this organism can also absorb things through little cracks that exist when these surface proteins line up to form this impervious barrier to the host's immune attack. It's a very complicated system. It's got lots of genetics associated with it that's just beyond the scope of this lecture. But just know that the proteins that are produced that coat the surface of this parasite all have certain things in common. They have a glycolipid anchor, for instance. The C-terminal domains of these proteins are basically similar. There's a region in the middle which is identical in every way. And then there are variants associated with the N-terminus. And that varies with each of these proteins. As you will see, there's a progression of these proteins as the infection progresses. And this progression is known as antigenic variation. And this parasite has become famous for lots of different studies trying to unravel exactly how antigenic variation occurs and what is the significance in terms of not just for the parasite and its life cycle, but also for the pathogenesis. This colorful representation gives you an idea of what the organism might look like to the host immune system. This might be an early form of the infection, which is started by the bite of the tsetse fly, and very soon thereafter, after these are all killed because the immune system catches on to the fact that they've now been infected with a green, let's make it a green organism. Unfortunately for us, the blue organism has already arisen, and killing off the green one simply leaves the blue ones to take the place of the green ones. I'm using colors here, but obviously they're not colors differences. These are antigenic differences, but for the sake of argument, we'll just use the colors. Once the antibody system catches on to the fact, the immune system catches on to the fact that we've got blue organisms now. They're not green anymore. We killed all those, but we've got some blue ones now to take care of. Where'd they come from? No problem. We can still do this. So the immune system launches an attack, kills off all the blue ones, but unfortunately, during the replication of blue parasites, a very few pink parasites arose because these are timed variations. we know their time because you can do this in vitro without antibodies, and you can still get the same variants to occur. The pink organisms then fill up the space left by the blue ones. No problem, we say, we can still make an antibody response against the pink ones that will kill all of them, but you know what I'm going to say next, because the next color is yellow. And if you had colors to represent, let's say, 2,000 different colors, that's basically the number of different antigen variants the trypanosomes can produce over their lifespan if they're called on to do that. And of course, they never get up to 2,000 because very serious consequences occur in the meantime as the result of having to deal with each one of these new populations of parasites over, let's say, a month or 2 months or a 6 months period. It's complicated in ways that relate to both the timing of when the variants are produced and the amount of antibodies that are that respond to those antigen productions. It's not important to know the exact timing of all of this. This is more of a research problem than a pathology problem for a clinician. But just know that in dealing with all of these, you have to be aware of the fact that if you wanted to make a vaccine, for instance, against this, it might be very, very difficult to try to choose which antigen to start with, because in essence, they can avoid virtually every approach that we've tried so far to make an antigen-proof vaccine, which nails all of these variants. It's just impossible to do it, and that's why we don't have a vaccine right now. And now Dr. Daniel Griffin is going to give a clinical vignette illustrating the pathogenesis of this infection.
This is a 42-year-old male refugee in Canada from the Democratic Republic of the Congo. This is an upper-class university professor of French in the Democratic Republic of the Congo. He had been, unfortunately, imprisoned, tortured, and then had fled and lived in the jungle for a few years, finally reaching a refugee camp in Tanzania and then moving to Canada. He came to the Canadian healthcare system 15 months after arrival in Canada. He was sent to psychiatry as he was felt to be unstable emotionally and was having delusions, hallucinations, depression, and what they felt were post-traumatic issues. He was under psychiatric care for about a year, but did not improve and was actually becoming worse. And he was sent to the hospital for evaluation by the medical team. He described a minor low back pain and is noted to be very somnolent, and he has fever. His physical exam is only remarkable for somnolence. On his labs, he's a little bit anemic. His basic tests were not particularly remarkable. HIV negative. His ESR, his erythrocyte sedimentation rate, was 60, which is elevated, markedly elevated. Normal, most labs would only be up to about 20. He did not have eosinophilia, but he did undergo a lumbar puncture, which was abnormal with elevated cell numbers. Radiology, he underwent a CT scan, which showed some adenopathy in the mediastinal, in the aortic, and the axillary under the arms there. His head MRI showed nonspecific midbrain abnormality, and there was diffuse edema. And when asked, this is something that we often do, we ask the patient what they think is wrong. He said, and my French, I should leave people to read this, but j'ai la maladie des sommeils. Now, a little bit about the progression. So we're going to be talking about the clinical presentation of African trypanosomiasis. And there really are two distinct forms, as Dr. Despommier has described for you. There is the form of African trypanosomiasis that we see in the eastern part of Africa, which is rapidly progressive, and the rhodesiense form. But then in West Africa, we're going to see a more slowly progressive, months to years progressive. And this person, as we note, is from West Africa. It's going to start off initially with a chancre, basically a site where the tsetse fly has bitten the person. Early on, intermittent fever, body aches, but then things are going to progress to ultimately to a neurological stage of the disease. Now, what are these two stages, as I started to describe? There are two stages to the clinical presentation for both both forms, East and West African trypanosomiasis. One is the hemolymphatic phase, where we have this painless chancre. Now, the actual bite of the tsetse fly, it hurts. These are quite impressive little vectors. The second phase, though, this is the real symptomatic phase. This is the central nervous system phase. This is where we have headache, depression, coma, and ultimate progression to death. Now, the difference, and this is important between the two types, your Trypanosoma rhodesiensis. This is rapid onset of CNS symptoms. We have a dysregulated inflammation, lethargy, coma, death. Now, West African or Trypanosoma gambiense, this is a slower development of CNS involvement. As mentioned, weeks, maybe years. We have a lethargic behavior for quite a while, coma, death, but often the death is due to pneumonia, interesting enough. So we're seeing quite different rates of progression. We're also seeing different reasons why people would succumb and die from these different infections. Now, this is a sleeping sickness clinic in Africa, and unfortunately, there still are areas where there are significant numbers of people affected by West African trypanosomiasis. The incidence of East African has actually dropped considerably. Now, what about diagnosis? Now, definitive diagnosis depends on finding the organism in either a blood smear, a lymph node aspirate, cerebral spinal fluid, or aspirates taken at the edge of the chancres. Now, cultures are going to be more sensitive than just these direct visualizations or smears. But what has really changed things has been a number of screening programs and a very simple serological screening test called a card agglutination test for trypanosomiasis, or CAT test. And we'll be showing a picture of that a little bit later. Molecular testing is another approach. It's still not widely available in endemic areas, but it's becoming more available as technologies have been developed that allow for isothermal nucleic acid amplification testing. So instead of PCR, where you need a thermocycler, electricity, these are simpler, more available technologies. Now, as far as examination of CSF, this is mandatory because, as you'll see when we get to treatment, we're making a distinction between early and late stage, and this is going to affect the treatment. So we use a very low threshold. White blood cell counts greater than 5 is enough to be indicative of CNS involvement and would trigger us to treat for that. One thing that is not common, but of interest, and I'll make sure we throw it in here, is that sometimes you can actually see large plasma cells containing eosinophilic inclusions. And these are Mott cells. They're uncommon, as I say, but they're characteristic. And the appearance is due to large amounts of IgM that these plasma cells are unable to secrete. Now, here we see a village, not your typical setting that people might be used to with regard to doing a lumbar puncture. I'll bring our arrow over. And right there, we're actually getting cerebral spinal fluid from this gentleman's spine. Everyone else is watching, enjoying the show. Early on in disease, when there are significant amounts of trypamasticotes in the bloodstream, sometimes you can actually identify these on a blood smear from peripheral blood. Now, over time, our diagnostics have gotten much better. And not only have they developed nucleic acid amplification testing, but they've also developed what really has moved things forward in a lot of parts of the world, but the idea of isothermal. So instead of needing that fancy, expensive machine that makes things hot, makes things cold, they actually have the ability to do nucleic acid amplification at basically the same temperature without having to heat it up. So this, as you see, the lamp is a type of nucleic acid amplification testing that's increasing the ability of this technology to be available in the areas where it's most needed. The card tests, I think I described before as a lateral flow, but I'll correct that because it's not a lateral flow. It's actually going to be using it on this card, and you're going to see an agglutination. Okay, and here we see the different levels of agglutination. Now, what about treatment? As I mentioned before, treatment is going to be based upon species and stage. In the early stages for Trypanosoma gambiense, So here we are in the west. Just think of where Gambia is. That might help you. This is over here. That might help you remember. And I think we need to rename this Zimbabwe, and that'll help people remember that it's over here. But early stages, pentamidine and suramin are two options. Pentamidine is preferred. Suramin has quite an issue with side effects. But suramin is the only drug option in East African early stage trypanosoma infection. Now, as far as late stages, Trypanosoma gambiense, the preferred is actually a combination of eflornithine and nifurtimox. Okay. Now, in some cases, eflornithine can be given as monotherapy, and an alternative is to use melarsoprol. Now, for late stage East African, the preferred is going to be melarsoprol. And here you see these different wonderful compounds, nifurtimox and eflornithine, suramin and melarsoprol. Now, what about our patient? Our patient had a blood smear which was negative. The lymph nodes were negative, but the CATT, the CAT test, was positive. And a PCR on the CF was done up at McGill, which is the reference tropical medicine lab in Canada. The patient was treated with monotherapy with eflornithine. And the patient did respond slowly, but improved over several months. And by 1 year later, he was reportedly almost back to normal. So this is a very nice outcome in this case.
So prevention and control remains our best way of dealing with this infection, rather than having to treat individual patients. Perhaps we can save large numbers by simply eliminating the vector or controlling the spread of the infection through reservoir hosts. So in East Africa, controlling the vector, the tsetse fly vectors, is relatively simple because of its relatively large distribution per fly. 1 square mile per fly seems, if you just simply slapped yourself, if you see a tsetse fly in your hand and you kill it before it has a chance to bite, you've now cleared 1 square mile of territory. It's quite remarkable, actually. There's a little bit about this biology of this fly that's kind of unique also, and that is that they don't lay masses of eggs. They lay 1 egg at a time, and the actual egg hatches inside the fly, and the thing that crawls out is the larva of this fly. And they usually lay their egg, or their larva rather, under trees and shady areas. It then pupates, and it can sit there for some time before it actually hatches. So they're difficult to control once they've reproduced, and once that stage is in the ground, it's probably impossible to find out where that happened. The thing that makes controlling very difficult in East Africa is the fact that almost all of the game animals harbor this infection. So the tsetse fly is an indiscriminate biter. It will bite cattle, it will bite people, it will bite hippopotami, it will bite rhino, if it can get through that tough hide. And as a result, virtually almost all of the tsetse flies in East Africa are infected. In West Africa, the situation is quite different. As I mentioned earlier, they breed along riverbanks, and so here's a very nice ecological shot of where you would find these tsetse flies. Not out in the middle of the rivers, of course, but the moment you docked your boat, if you hadn't cleared this area of brush and trees on either side, you would run a very high risk of being exposed to hundreds, if not thousands, of tsetse flies, which would descend on you the moment you stepped off this barge that took your car across this river somewhere in West Africa. That's a very typical situation there. So the way to avoid this infection in West Africa is to build your cities and large villages away from the banks of the river. And of course, that creates inconveniences too, because water is a very precious thing, and everybody needs it for not only irrigating their crops, but also for drinking purposes. So any way you look at this, controlling tsetse fly biology or controlling the infection in a reservoir host has not resulted in the eradication of this disease in too many places. If you look at the distribution, however, you now have an explanation as to why a lot of people from Europe moved to South Africa and not to equatorial Africa, because the tsetse fly belt ends slightly north of of South Africa. There are lots of other topics to be discussed relating to the control of African trypanosomiasis, and I've selected a good review article for you in this. It's a recent article, Past and Ongoing Tsetse and Animal Trypanosomiasis Control Operations in 5 different African countries, and it's a very long review and a very thorough review, and I highly recommend that for you. We've got many episodes, 5 as a matter of fact, on our TWIP podcast to listen to in various aspects of trypanosomiasis and the molecular biology of it and the biochemistry of it as well too. And you can find those podcasts at microbe.tv/TWIP. So next time we're going to discuss the yang of the trypanosomiasis twins, African trypanosomiasis, American trypanosomiasis. What's the difference? How do they vary in their biology, in their pathology, their transmission, etc.? So thank you for listening, and look forward to talking to you next time.